Thank you. So thanks to Tari for inviting me to this workshop, which is very broad in interest. But still, even within this broad interest, I think my talk will be a little bit of an outlier. So it's probably a good idea to interrupt me if there's something unclear during the talk. And I'll tell you about exploratory adaptation. And I would like to tell you about, it will have two parts. The talk will have two parts. The first is an experimental part. I will review experiments that show what is exploratory adaptation and what is it characterized by. And then the main part is a theoretical part that will describe a model that we have developed and are working on, a model for exploratory adaptation, which relies on random networks. But before that, because adaptation is a very widely used word and it can have different meanings, I will spend a couple of minutes defining what I mean by exploratory adaptation in a rather general context. So we think of adaptation in terms of a cell that is subject to some external challenges. Okay, for example, temperature, high temperature, for example, mechanical stress or other types of signals that constitute some kind of a challenge to the cell. And the cell adapts to it. And there are many such possible challenges. And very often to these kinds of conditions that you can see here, cells have developed over evolution adaptation programs or physiological adaptation programs to deal with them. So what usually underlies these physiological adaptation programs is some kind of a regulation system. And a large fraction of systems biology is all about understanding these control regulation systems. They often have some kind of input, then some kind of chain of molecular event that we can understand, and then finally there's an output which constitutes the adaptive output or the adaptive response of the cell. And there are many such adaptation programs, physiological adaptations. Cells are very flexible, they can respond to different conditions, but ultimately this is a finite repertoire. So the capacity to respond in this way or to adapt by physiological adaptation is ultimately limited. Even if it's a large repertoire, ultimately it comes to an end, and comes the day when the cell is confronted by an unforeseen challenge. And an unforeseen challenge, for example, an unknown toxin, a new type of antibiotic, is one for which the cell does not have an adaptive program ready to invoke. And this can be, for example, a large genome rewiring or some other kind of toxin, something that has not been encountered along the history of the cell, and therefore there is no ready-made response. So what would happen then? And one possible scenario is adaptation at the population level. So as we know, cells are variable in all of their properties, both genetic and phenotypic. And some cells may be by chance more adapted to the unforeseen challenge, whereas others will die out during this selection process. And this is, of course, evolutionary adaptation. And actually, in some communities, this is the only definition of the word adaptation that is used. But, however, there is another possibility. And this possibility is that individual cells may adapt to unforeseen challenges, meaning that although they do not have a prepared adaptive response in physiology, they will nevertheless succeed in accommodating this unforeseen challenge. And this can be done by harnessing their inner complexity, which is here represented by some kind of many-body system inside the cell. We'll say more about that soon. And if they can harness this plasticity, this internal complexity, they may adapt at the individual level to unforeseen challenges. Okay? And So here comes the first part. Now we'll tell you about the experimental part. And this is a collaboration with ELSC from the physics department at the Technion, long time collaboration. And one of the major ingredients in this experimental paradigm that we've developed is how to apply this unforeseen challenge. Okay, so our goal is to confront cells. These are experiments on yeast cell populations. Our goal is to confront the cells with a challenge that they had not encountered before. Okay, and how do we do this? Without going into too many details about the genetics of yeast, the gene regulation system of yeast is composed generally of different modules. Each one has a more or less well understood goal or role in the cell. And here are two modules schematically represented, two modules of metabolic function in the yeast. One of them has to do with sugar metabolism, the other has to do with amino acid metabolism. And each of them is a kind of a stereotyped control system, as I described before. This one responds to the external sugar in the medium. This one is invoked in the case of amino acid starvation. And under normal conditions, although they're part of the entire cell system, the entire cell network, they are weakly coupled to one another. And typically, if you give some local perturbation, such as changing the sugar, you will see this module being active and responding. And if you take out the histidine or whatever from the medium, you'll see this module active. And so the response is relatively local. Now, what we do to apply a challenge and to perturb the system very strongly is we couple these two systems directly, meaning that we choose arbitrarily one enzyme from this amino acid system, amino acid synthesis. Instead of having its own controls and its own regulation, we rewire it to receive control from the other module. Okay? So now this particular his3 enzyme or whatever is being turned on or off by a regulation system, by a promoter that actually belongs to a different part of the regulation system. All right, so this is the perturbation, and actually what this does is it has the potential to ruin the entire logic of the system. Why? Because we have here a switch. Okay, this is the regulation of the sugar system. I can turn it on by putting the lactose in the medium. I can turn it off by putting glucose in the medium. And then this will turn on an enzyme that will, a gene, sorry, that will create enzymes that have to do with a completely different function. So it's responding to the wrong stimulus. It has the wrong dynamic range, inappropriate dynamic range. And basically, for example, if I put the sugar that represses this control system, and I demand this to be produced by taking out the histidine from the medium, I'm confronting the cell with complete frustration. It has the gene, but it cannot produce the appropriate enzyme. Okay, so this way I have tools in my hands to confront the cells with a very strong perturbation that they have no way of dealing with. Okay, there is no pre-evolved response or physiological adaptation that can answer to this challenge. Okay, and to make things worse, I have another handle on the severity of the challenge by a competitive inhibitor, a chemical inhibitor, that I can apply to the medium. And in that case, even if there is a way to create these enzymes, there's something that's blocking them in the medium, which makes it even more difficult. Okay, so now I take these cells and I grow them on plates, and I would like to see what they do, what happens to them. So in galactose medium, where the switch is on, the challenge is not so severe. I mean, not exactly the same dynamic range maybe, but cells are growing colonies, and this is how yeast cells look after a couple of days in place. Okay, there's many colonies. However, after the same couple of days, when the switch is off in the medium, there are no cells at all. Okay, 2 days, no cells. 4 days, no cells. 6 days, start to appear the first colony. And this is after 14 days. And the reason why there is such variability is because the time of the development of these colonies is also extremely variable. So this has already the flavor of some adaptation that's taking a long time, a variable time. However, you can see that there's a large fraction of cell that ultimately were able to produce colonies here. Okay, so this is more or less has the first signs that this is what we were looking for. And it's clear that this is a dynamic process. Right? It's taking them time, a long time, and a variable time. And a way to follow the dynamics of this process is to grow the cells rather than on plates, to grow them in continuous culture. And continuous culture enables me to follow the population density a long time. You can see here, this is the time in hours. This is a very long, these are long experiments. They take quite a while. And if you see here, if you follow this gray curve over here, this is the population density, starting at the high, then a high value at time 0. Now I apply the unforeseen challenge of switching the medium to the off switch, and you can see that the population decreases. There's a collapse of the population following the unforeseen challenge, but there's a recovery after a while, and then you can see the dynamics of this adaptation process in terms of the population density. Okay, so this is the analog, the continuous analogs of the plate as you've seen before. But now I can use this chemical inhibitor to increase the severity of the challenge, and what you see is a qualitatively similar curve, but takes much longer, and the population collapsed to a much lower value. Okay, this is actually the floor of measurement. Most probably the population collapsed much lower than this value, but this is already noise. So it's taking a longer time if the challenge is harder, is stronger. Okay, and this also gives us the impression that what we're seeing is really a response to this challenge that we confronted the cells with. So what's going on in the inside of the cell? If we want to look at the gene expression of the cells, yeast provides us with a lot of technology to follow what the cells are doing. So we can, basically we sample the cells a long time in this continuous growth, and then we can map out what genes are expressed and to what level at every point in time. And although, remember that the perturbation was supposed to be local, on the face of it was local, I took one gene and rewired it to a different place. Despite that, what we can see, you look at, this is a way to present as a function of time, and these are the genes one on top of the other, and this is the scale of upregulation or downregulation, expression or repression of genes. And here you can see a couple of clusters as a function of time. And you already see from here, from this picture, even qualitatively, that the response is quite local. Okay, so although we did the perturbation locally, a lot of genes are responding. And actually, there are hundreds of genes that are responding during the adaptation that you've seen before. And we did some clustering analysis. It turns out that you can more or less group them into two clusters. Some of them are repressed and then relaxed, and some of them are strongly induced and then relaxed. But there are a lot of genes participating in this response. And now if I go to the stronger perturbation with the chemical inhibitor, you see that the response is even stronger. The colors are stronger, meaning are brighter, meaning that the magnitude of the response is stronger. Actually, the clustering is also stronger. And what happens in this global response is that the natural or the original modules of the regulatory system are completely broken down, meaning that in each one of these clusters appear genes that have nothing to do with each other, nominally. On the other hand, modules of genes that are thought to be functionally related to one another divide between these two clusters. And so something breaks down in the logic of the system. And actually, it looks like this is a completely different kind of response. Okay? It's global, it's sensitive to the level of stress, and it takes a long time and a variable time. And the last thing I want to show you from this set of experiments is what happens when we repeat the experiment again. So now, I start from an ancestral yeast cell. I do the experiment all over again. And what you can see, this is the population density that we've seen collapses and recover. And these are two separate experiments. This is where the perturbation is applied. And qualitatively, the graphs are similar. The time is slightly different, but more or less the features of this adaptation are the same. However, if I go again into the gene expression and I measure the fold change of each and every one of the several thousand of these genes, I can plot them in experiment 1 as a function of experiment 2. So each dot in this plot represents one gene, and on this axis you see what that gene, for example, the gene that's over here near 3 has upregulated its expression threefold in the first experiment. However, in the second experiment, it has downregulated its expression. And the fact that these points are all scattered in a cloud have a very significant meaning, and that meaning is that in every repetition, the details of the genetic response is completely different. Okay, there's no correlation between what the genes do in experiment 1 and what they do in experiment 2.
This
Yes?
is also true for different populations on the same plate?
Say it again?
This is also true for the different populations on the same plate?
Yes. It doesn't come out of this experiments, but yes. Okay, so this is the summary of the experiments. So one thing is, yeah, more questions.
Is there a proof that adaptation the individual level and not evolutionary adaptation?
Actually, there's a lot of experimental work towards this question that I'm not going to cover here. Okay, I'll give some references, and if you're interested, there's a recent review by Ayers Brown which covers all of these experiments, practically 10 years of experiments, and they are significant. But the short answer is yes. It's not a population level selection. And okay, so the first feature that I will need to address is that many cells adapt. Actually, not 100%, but a large fraction of the cells adapt. The second is that it's a dynamic process that takes time, can take relatively long and variable time. The internal degrees of freedom are responding in a global and unrepeatable manner. And it's breaking down the modular logic that usually exists with small perturbations. And the response is sensitive to the level of stress. Okay, and these are some other references. There's also other experiments in flies that are related to this phenomenon. And this is by now pretty well documented experimentally. So now you can go to two directions. One direction is related to what has been asked here is the mechanism of these adaptation processes. But what I would like to focus on today is some organizing principle to start to inquire what kind of organizing principle, theoretically even, can underlie such an adaptive process with this kind of features. Okay? So what I will suggest is a model that implements the organizational principle of drive reduction. And drive reduction can be considered a primitive form of learning without computation or information transmission or processing. And here's the algorithm, quote unquote. It's not, hardly can even be called an algorithm. The principle of drive reduction can be formulated as follows. So stress induces some random exploration in the internal degrees of freedom of the system. And this exploration occurs in some space of random, of possible configurations. As long as stress is high, keep searching and kind of randomizing your degrees of freedom. And when a favorable configuration happens to come along, the stress will automatically be reduced, and then the exploration will be reduced with it, and hopefully the system can stabilize. Okay? And this has been studied a while back by in Shimon Morom's lab with ex vivo networks. It was implemented by a semi-synthetic system that interacts with a computer, and it was later discussed in the context of cell biology by the group of Yoav Soen at the Weizmann Institute. And what I will show you today is, as far as I know, the first computational implementation of this principle. Okay, so here are the computational details. We're thinking of a large number of internal degrees of freedom, microscopic variables. We're thinking of nonlinear interactions, and we're utilizing here a very popular model of neural networks. I think in this audience, I don't even have to introduce the model, right? It's been introduced many years ago, and actually it's also a variant of this model has also been used to describe gene regulatory networks, like Kaufman, Wagner, and others. And in this equation, W is the interaction matrix, and phi is a saturating function that keeps the system from diverging. And a lot is known if W is a Gaussian random matrix. There's a lot known about this. In particular, there's a transition from dissipative to chaotic motion. And more complex networks that have structure in them and have correlations in their elements are just recently beginning to be studied, and several references are given here. But if I go back to our use of this system, typically we're looking at, I'll say a lot more in a minute about the properties of this interaction matrix, but in general, I'd want to be working in a regime where the dynamics are irregular. Okay, typically irregular. So I have typically irregular dynamics, but everything, these degrees of freedom, the microscopic interacting degrees of freedom, do not determine directly the functionality of the system. There's some buffering. There's a macroscopic level of description, and the phenotype is some combination of internal degrees of freedom, which is degenerate. Okay, so the idea is that multiple gene expression patterns can support the same phenotype. And so this is represented very simply by a linear combination, and I call Y the phenotype of the system, and that is what interacts with the environment and somehow has to comply with an external constraint. Okay, so there's interactions, there's microscopic degrees of freedom, but there's also a variable that represents the functional aspects of the cell, and this is a low-dimensional constraint that this has to satisfy. Okay, so what does it mean to satisfy? If this is the desired or the required value, then there is some level of mismatch or stress that the system is going to feel, but it's not precise. So it can be somewhere around that area. So this is an additional layer of approximation. There's not a very strong optimization problem. It can be somewhere in this region, but if it deviates from a certain finite region, then the stress is going to kick in and something's going to happen. And what's going to happen, there's going to be this exploratory dynamics, these random dynamics. Okay, so this is the function that governs the exploratory dynamics. If the mismatch is low, there's going to be no exploration. But once it deviates and the mismatch is high, then internal degrees of freedom are going to start wandering around. And now I have to specify how they wander around, right? What are they going to do in order to search for a new configuration? So for this, I imagine that the W matrix, the interaction matrix, has two parts. It has a topological backbone, and it has a connection string. Okay? And I assume at the moment that the topological background is fixed, and the connection strength just slowly, randomly wander in the space of connections by a completely independent random walk, each connection. Okay? So this is the summary of the model, and it's really a very primitive algorithm. And so, okay, so this is the equation of motion. This is the structure of the exploring degrees of freedom, right? The connection strength. They're slow relative to the internal dynamics. And I can have two situations. If the stress is high, the mismatch is high, something is random walking in this space of connection, and then if everything relaxes, then there's going to be no change. Yes?
So J is a Wiener process?
Yes. It could be different things as well. I can tell you more details later, but in the implementation that I'm showing now, it's a Wiener process. More question? Yeah.
Is this somehow implementing a kind of a local search algorithm, or is it it's purely random?
Okay, so all the connections, in principle n squared connections, but I'll show you that it's not n squared, but all connections in the matrix are independently performing a random walk. So this is a random walk in 1,000 dimensional space, okay? And the main question is, can it converge to satisfy the constraint, and if so, under what conditions? And of course, it's not clear a priori that it can converge. And if you just put this random, nice random Gaussian ensemble that we're used to seeing in most models, it's not going to converge. At least we have not seen it converge. If you think there may be too many connections and you take a very sparse Gaussian random matrix, it's not going to converge either. And so at this point, I'm going to state the main theoretical results of the talk. And this is one, convergence to stable states satisfying the constraints is possible. And second, it's not universal. It depends on certain properties of the matrix of interactions. And we have identified at least some properties that it depends on. And the primary pattern, primary property is the pattern of connections. So homogeneous patterns of connections do not converge, but heterogeneous patterns promote convergence, as I will show now. Any questions further?
Yeah, Maureen?
Yeah.
So do you not have to assume that your diffusion process stays bounded somehow?
Yes, let me go to the next, you'll see this in the next figure, yes. Is it the same question?
If I get you correctly, coordinates in your vectors correspond to particular yeast cells in your example, right?
Sorry, say it again?
If I get you correctly, coordinates in your
No, coordinates correspond to, okay, so thanks for the question. All of this describes what goes on in a single yeast cell. Okay, so this is the genetic network. I describe everything that goes on in a single yeast cell, and the goal is to describe an adaptation that can occur within the lifetime of the cell without having a pre-designed system that deals with the current.
So what does a matrix entry correspond to in your model?
It corresponds to the genetic regulatory network. Okay, so let me go to the next picture that will explain the problem of divergence. So this is an example of what convergence looks like. Okay, so I start the system and t equals 0, and there's this corresponds to the Y star. This is the desired value or the constraint on the phenotype. And the phenotype, which is a combination of degrees of freedom, fluctuates, while the internal microscopic degrees of freedom fluctuate even faster. And on the slowest time scale is the change in these connection strengths. This is a really slow process. However, it doesn't have to go very far in order to find a solution spontaneously. Okay, so this is what the connection, this is 4 out of the 1,500 potential connection strengths. This is less, but we'll talk about that in a minute. But these are 4 out of the many connection strengths. See what they're doing? They're just slightly jiggling around, and then they find a solution which satisfies this constraint to the desired accuracy, and then the system locks on a fixed point. Okay, so this is what convergence looks like. And what does it depend on? So as I said, it depends on the properties of this matrix W. And in order to study how it depends on W, what we've done is the following. We've created a set of topological ensembles and repeat the simulation many times for each ensemble. And then we plot here. Each one of these lines corresponds to an ensemble that I will explain in a minute. And the bar shows you what fraction of the simulations manage to converge to a stable state that complies with the external demand. Now, the initial conditions are randomized. The constraint is randomized. It's completely arbitrary. Everything that's common in each of these lines is the distribution from which connections are drawn. So, for example, the top line, and as you know, in any graph, the gene regulatory network is a directed graph, so we have a distribution of outgoing connections and in principle separate distribution of incoming connections. And so, for example, the top line shows what happens if you take W to be a matrix drawn from, with connections drawn from a scale-free distribution. Okay, scale-free distribution means there's a power law. The actual accuracy of the power law doesn't really matter much, but you need a very broad, heterogeneous pattern of connections. All right? So if you take the incoming and the outgoing, we draw them out of a scale-free distribution, you get about 70% convergence for a fixed time window. Now if you take a scale-free out distribution and an exponential incoming distribution, you get about 60%. Now this comes an interesting fact. If you switch them around, if you take the transpose matrix, right, and you draw the outgoing connections from an exponential distribution and the incoming connections from a scale-free distribution, this drops by a factor 4. The convergence is much less. And the same you can do with a scale-free binomial, gives you about half convergence. Binomial scale-free gives you an order magnitude less. And this tells us that this is not a property that relies on the spectrum of the matrix W, as some properties do rely on the spectrum, but this is not a spectral property because the spectrum of these two types of matrices are the same, okay? So there's a dynamic process that is really sensitive to the distribution of outgoing connections, and the most important property that you need for a considerable convergence to happen is a very broad out-degree distribution, right? Now, if we go back for a minute, somebody asked, what is this network? And I go back and reiterate that the network is supposed to represent the gene regulatory network of yeast.
So,
Yes.
so convergence means a fixed point also?
Yes. It can also, all right, let me go back and say, it can be a fixed point. It actually can also be a small limit cycle
Okay.
that lives inside of this constraint. Okay, so sometimes we will see limit cycles, but sometimes, very often, we will also see fixed points. All right? Okay, so if I go back to the question, what does this matrix represent? And I go to the literature and I look at distributions of gene regulatory networks that have been studied a lot, what you see here is two graphs of this. These are the incoming connections. Here you see the distribution, or the number actually, of regulating proteins. So how many proteins am I regulated by in the regulatory network? And this is a semi-log plot showing that this distribution is rather exponential, rather narrow. But on the other hand, the outgoing connections, right? How many regulated genes am I regulating, right? This distribution, this is a double logarithmic plot, and this is more or less a scale-free distribution, a power law. And in any case, there are very strong hubs. There are very large hubs in this connection pattern. There are regulatory genes that can regulate hundreds of other genes, and they are called hubs in the network. And this is what characterizes this broad distribution. And so out of all these ensembles that we have tried, the yeast regulatory networks actually sits here, and this is quite encouraging. Okay? So this is a property of gene regulation. It's not a very typical, maybe, network, but this is a property that is in line with what we know about gene regulation. Okay, so now we can study how it depends, how this phenomenon depends on different parameters, for example, network size. So it turns out that if the network is rather small, convergence is pretty high at 80%. And the difference between the two types of, two families of ensembles, the one with the broad outgoing connections and the one without the broad outgoing connections, becomes more pronounced as you increase the network size towards the sizes that are typical. So we're talking about typical sizes of networks of 1,000 to several thousand. This is the regime that's relevant in terms of the cell. So you can see that there opens up a really big gap here between those systems that have a broad outgoing degree distribution and those that do not. The other thing you can look at, so we've mentioned the concept of hubs. What you can do, which is typical analysis of this kind of networks, is you can ask, what happens to the convergence fractions if you start to delete out nodes out of the 1,500 size network? So if you just take any random node out of the network, this is around the 60% or 50-something% convergence, it doesn't change by much. But if you, on purpose, pick the hubs out of the networks, you can see that the convergence fraction decreases. So the hubs play an important role in this dynamic process of convergence. And this is actually a different analysis that highlights a related property. If you now take many, okay, so within this ensemble of scale-free matrices, there are differences because it's a heterogeneous distribution. There are differences between realizations of the matrix. And here what we've done is grouped them according to the largest hub. So this realization happened to have the largest hub of a size 200. And this realization happened to have one of a size 1,000. And you can see that also this is correlated positively with the convergence fraction. So again, the hubs play an important role in this dynamic process. Okay. Another important parameter is this is sometimes called the rate of exploration. And there's a parameter in the model that D, which is sort of the diffusion constant of these connection strengths. And what I've shown you until now is a D of about 10 to the minus 3. What we've done in this graph is explored how sensitive is the convergence to the value of this diffusion constant. So 10 to the minus 3 represents the fact that diffusion is 1,000 times, typically 1,000 times slower than the internal dynamical timescale, which is in these units 1. But you can actually change this over quite a broad range, several decades. You can change this between 10 to the minus 4 to 10 to the minus 1, and still get a considerable, between 0.2 and 0.7, something like that, of convergence fractions. Until over here, it really collapses fast, and this is where the timescales are not separate anymore. This is where the exploratory search, overlaps in timescale with the internal dynamics, and there you cannot get an effect at all. And if you can't see this, so I'm just emphasizing that there are 8 decades in this graph on the x-axis. Okay, so the diffusion constant is changed over a very broad range. Okay, and what else we can do? We can think about what happens as we increase the severity of the challenge. So there are at least 2 ways to do this. One of them is to make this comfort zone inside the mismatch function smaller and smaller. And you can see in this graph, this is the epsilon, the width of the comfort zone. And as you decrease epsilon to be very, very small, this effect collapses rapidly to the earth. There's no convergence. So in some sense, this exploratory adaptation depends on the fact that we don't push the system to have one particular minimal value. And in this sense, this problem is probably a little bit different from standard optimization problem. But the fact that we are allowing it some room, some space to be in, is actually what's allowing this exploration to succeed.
There's a unique fixed point?
No. There is not a unique fixed point. Okay, every time we run the algorithm, the microscopic degrees of freedom find a completely different configuration. Okay, there are many fixed points. The network space is dense with fixed points. Okay, and maybe this is where I should put up this graph. Okay, so this is a question now about if the system has found a fixed point, there's got to be a fixed point of the open loop dynamics, right, of the original dynamical system. And what you see over here is just a simulation running an ensemble of matrices W without any feedback and without exploration, just to test how many fixed points are in the network space. And you can see that these narrowly distributed connections matrices have very, very small number of fixed points, and they actually go to 0 as the network size increases, and this is well known. These are also the matrices that are homogeneous. They have no fixed points as you increase the network size. However, the scale-free networks seem to be saturating at some intermediate value, which means that the space of this is a much larger n. This goes to 10,000. Of course, we don't know what the asymptotic value is, but we can see that within the range, again, within the important range of several thousands, there's a big difference between those networks that have broad outgoing connection patterns and those that don't. And the colors show you that this hierarchy over here is exactly parallel to the hierarchy of the convergence fractions under exploratory adaptation. And this, to understand this, is actually an interesting problem that we're working on now. The last thing I want to show is what is the distribution. Until now, we've looked at convergence fractions at a fixed time window. Now, if you take very long time windows and you ask, what is the distribution of times? Remember, in the plates, we saw that the cells adapt over very broad time scales, or distribution of time scales. And this is what the distribution of time scales looks like. This is probability density, and here is a time. And if you take a look at this, for example, this dark green curve, this is one of the higher fractions of convergence. And this is a semi-log plot, and it can be fit by stretched exponential. So this is a kind of a bizarre distribution, not very well understood. It has a broad CV larger than 1, and it can be described by a stretched exponential. Actually, all the networks that have converged with some fraction show the same type of distribution of convergence times. They all look like stretched exponential. And this, just using free association, this reminds us of some complex systems, complex dynamics that happen in high-dimensional space that also show these kinds of distribution of times, like, for example, glasses. Okay, so the last slide, I want to address a really difficult question. And if we go back to the first thing we talked about, which is the pre-design, so to speak, or the physiological adaptations, and what I've shown you today is the exploratory adaptation. One of the major questions when you look at a biological system is, how do these two properties, completely different types of responses, how do they survive and coexist in the same system? And one may even say that this is a kind of an exploration-exploitation balance, because if you have well-prepared control systems for adaptation, then you're exploiting what you've encountered before. But if you have some power to explore, then you can confront the unforeseen challenges. And what we think is happening is that there are some properties of these systems that actually support both types of adaptation. So for example, in the yeast cell, like I said in the beginning, there's a lot known about modules, about genes that are regulated in concert. Sometimes they belong to the same function. Sometimes they constitute some sort of time-ordered gene expression that is required. And so there's a lot known, and there are similar, this is taken from the yeast genome, and there are master regulators that control specific programs of gene expression. But at the same time, if you kick the system strong enough with an unforeseen perturbation, you find the same module can play a role even statistically. If the entire system is undergoing some global dynamics, the fact that you have these kind of structures, even without knowing what exactly the genes are, can give rise or can support some general dynamical properties that we think we're seeing in this exploratory adaptation. And with this kind of speculative, I will end and summarize. And so I've shown some properties of exploratory adaptation in experiment, and we've talked about that. And then the main part was developing a random network model that represents what happens inside a single cell and how the cell can harness its own internal complexity, a lot of degrees of freedom that are interacting, and some feedback from the environment in order to adapt in an non-repeatable and unforeseen way. And we've seen that convergence is possible. It's not universal. It depends on properties. There may be other properties that will promote convergence that we haven't seen yet. And these are the main properties that allow this kind of convergence. So with that, I would like to thank Hallel Schreier, the PhD student who did most theoretical work, and Yoav Soen is our collaborator on this. And this is Haifa, if you're interested in visiting, and this is the Network Biology Research Lab. And I would like to thank my fellows at the Network Lab, which is a really nice environment where conversations with many of these people have provided the inspiration to this work. Thanks.